All right, great. So why don't we get started? Thank you everyone for joining us. I'm Paul Danis with the consortium, and we're excited to have Mohammed Zenaidee today with us. Tell us about some of his interesting work in native top-down. Let me first give a couple of little thank yous. First off, to our sponsors. Very grateful for them to give us the support we need to run these kind of activities and grow the community. Thermo Fisher Scientific, Brooker, SciEx, and Lilly are our major sponsors, and we also have support additionally from Agilent, New Objective, Pfizer, and Nautilus Biotechnology. So we're grateful to them. If you see these folks around or if you're online, we really appreciate it. And if you have other people who may be interested in sponsoring us, please get in touch. We'll be happy to have a discussion around that and see how we can work together. Going forward, we have a couple things coming up. We have Ana Marinho from Brazil speaking in August. And September, we're still working on that. And then in October, of course, we have our annual vendor showcase where you can see all the cool new tools that everyone can play with. So with that, I'm going to hand it over to Inga from the University of Wisconsin, who will introduce Mohammed and run the rest of the seminar. Thank you.
Hey, thanks, Paul. Yeah, it's my great pleasure to introduce Mohammed Zenaidee. So Mohammed is currently the mass spec manager at the Australian Proteomics Analysis Facility. He received his PhD from University of New Wales, working with William Donald, and then he moved to UCLA in US to work with Professor Joseph Liu, who's here to support, as well as Rachel Liu. So Mohammed focused on the top-down mass spectrometry and proteoform characterization that he learned in PhD as well as during his postdoc. So his current research focuses on the fundamental mass spectrometry through investigating these electron spray ionization mechanism and the protein dissociation pathways, as well as method development to enhance protein characterization by mass spectrometry. Here we go, Mohammed.
Cool. Uh, all right. So before I start, can everyone see my screen and can everyone hear me? Cool. All right. So thanks for the introduction, Ing. It's great to be presenting some of the work that we've been doing at the facility, as well as some of the work that I'm quite interested in. If I sound a bit tired, I will just make a note that it is 1 a.m. currently for me right now. So please wake me up if I fall asleep. So basically today I'm going to spend a little bit of time talking about how we can use internal fragments and ion mobility for the characterization of disulfide bonded proteins. So a lot of the work that I'm going to be showing today will be using these types of techniques in order to take a protein, unfold it, and then use electron capture dissociation to reduce those disulfide bonds, followed by collision-induced dissociation to then fragment the protein and hopefully give us information about the structural characterization and be able to structurally characterize these proteins. So probably preaching to the choir here, but as we all know, disulfide bonds are crucial for maintaining protein structure. A lot of these disulfide bonds allow proteins to function and maintain stability. And they're a challenge to analyze in top-down experiments because these disulfide bonds can limit protein fragmentation. So if you take this protein, for example, and you fragment at this site, you can see that because it's still connected via the disulfide bond, you're actually not able to detect that fragment, which really limits your protein sequencing experiments as well as your protein characterization experiments. So in a lot of the work that I do, we do native top-down and intact mass spectrometry, where we take an intact protein detected within the mass spectrometer, isolate and activate it within the mass spectrometer to generate fragments. So something that we do differently from a lot of other places is we also include internal fragments in our analysis. So internal fragments are fragments that contain neither the C nor the N termini, And we found that we can include these fragments in order to extend the amount of information you can get on the protein from the protein backbone to give you better characterization on the protein, as well as understand the protein sequence a little bit better. So to give you an example, here's some data I collected from Joe's lab, or it might have been Carter or Ben, who I'm not sure are on, but this is an ECD MSMS of apomyoglobin. It's broadband, and you can get a lot of different fragments. And I'm going to be showing a lot of these plots. So these plots here are the fragments that are identified and matched to the protein. So if each of these fragments start at the ends of the plot, they are terminal fragments, and fragments within the center, which do not cover one of the termini, are internal fragments. So as you can see, the inclusion of internal fragments can help extend the amount of sequence information you can get from the protein, which gives us better ways to understand this protein sequence. Internal fragments have been shown to be quite important and can be used to look at disulfide connectivity. So this is some data that Ben collected a while ago for beta-lactoglobulin and lysozyme. And here they do electron ionization dissociation, and they generate a lot of different fragments and a lot of different internal fragments. And as you can see, by including the internal fragments, you can actually start mapping portions of the protein that is covered by the disulfide bonds. The disulfide bond connectivities are linked based on the color. So you can see that there's a disulfide bond link here, and there's a disulfide bond link there. And for lysozyme, you get a lot more disulfide bonds being formed. And by including internal fragments, you can actually extend the amount of information you get within the center of the protein, as well as give you information about the disulfide connectivity. However, this experiments were done on an instrument with electron ionization, and not everyone has that, and we didn't have one at that time. So what we do have is the cyclic ion mobility, and the cyclic ion mobility is a great instrument because it lets you do some very nifty things. So we do have access to collision-induced dissociation within the trap and within the transfer, the ion mobility cell, and the ability to do electron capture dissociation either before the ion mobility or after the ion mobility. So for this particular sets of experiments that I'm going to be showing shortly, what we're doing is we're getting our protein to come in, we're unfolding within the trap, doing ion mobility, doing electron capture dissociation to then reduce those disulfide bonds, and then doing fragmentation before detecting the fragments and piecing it all back together. Here's a picture of our instrument in our lab. It's fine. It probably could have been a cleaner picture, but it is what it is. Just in case people are not too familiar with collision-induced unfolding, I thought I'd give a little bit of a brief crash course. So if you take a intact protein, introduce some small activation, what you can do is unfold that protein without fragmenting it. So now you can see that that unfolded protein has a much greater collisional cross-section than the folded protein. If you put that into your ion mobility tube, or in our case, our cyclic ion mobility cell, and do ion mobility, you can see that you can separate these two proteins based on the collisional cross-section, and your more compact comes out earlier, and the unfolded one comes out slightly later. So basically, we'll be monitoring how we're unfolding the protein using the ion mobility. So in the next set of experiments that I'm going to be showing you, what we've been doing is we take our intact protein, all in native or in water, and we detect the intact protein within the mass spectrometer, unfold it slightly, do electron capture to reduce the disulfide bond before fragmenting it. So all the fragments that we identified that cross the disulfide bonds actually have this reduced disulfide. So the first thing that we wanted to look at was, can we use these types of internal fragmentation with collision-induced unfolding and ECD to characterize something like bovine serum albumin? So it is a common test protein. It's 66.5 kilodaltons and has many, many disulfide bonds. This is just our test sample, so we're just seeing if it's even feasible. So what I'm plotting here on the left is the collision-induced unfolding. So these are all the different conformational changes for BSA at different drift times. And on the right here are the fragmentation, the fragments that were generated from this experiment. So if we take a look at the first step where we barely unfold it or don't have any unfolding energy, you can see we get some fragments that are forming, but a lot of them are from the termini and only crossing very limited amounts of the disulfide bonds. So for all these experiments, we do have the ECD on. And if you increase your unfolding energy, you can see that you start getting more and more fragments, some that cross more of the disulfide bonds. And as we go further, you get even more of that happening. So basically what I'm trying to show you here is that as we slowly unfold the protein, we get access to more and more of the disulfide bonds, which allows the reduction of the fragments of the disulfide bond, which can then generate fragments that we can detect. Now, all the fragments here do have all the disulfide bonds reduced. So for example, if you've got a fragment that crosses 4 different disulfide bonds, you can see 4 different disulfide bond reductions in that experiment. But really, one of the main reasons we're doing this is we do a lot of work with venom proteins. So venom proteins are highly disulfide bonded, which can really limit characterizations of these proteins. They're a complex cocktail of bioactive proteins and peptides, and venom compositions can vary drastically between and within species of snakes. Characterizing intact venom protein remains a challenge, mostly because these venoms are very highly disulfide bonded to maintain structure and not very well characterized. So the first set of data I'll be showing would be from the forest cobra, Naja melanuca. And for these types of experiments, I'm only going to be showing the top-down experiments, but there's a lot more that happens to it. So the way we do these types of characterization is we get the whole venom, we do size exclusion chromatography to separate it all out, and we do bottom-up proteomics initially. So we do just a trypsin digestion to get you all the proteins that are in this venom cocktail, as well as we transcriptomics of the venom glands, because a lot of these venoms tend to not be well characterized. So we need to generate a faster library that we can search against. So we do transcriptomics to get us our protein library, which we can then match with our bottom-up proteomics to give us all the toxin families. And then from there, we can actually do proteoform profiling. So we can actually look at individual species, individual proteins within that complex mixture in order to understand the makeup of this better. So here's some data that we'd collected previously for this particular snake venom. And this is a PLA2 protein, and we can isolate the 8 and 7 plus. We get a lot of internal fragments, which can then be matched back to your protein sequence. This is the first time that this protein has been characterized by top-down mass spectrometry. But we know that these proteins are white disulfide bonded. So we wanted to see if we can then extend this further. So here are the main proteins that are present in that venom cocktail. And for the next set of data that I'll be showing, we'll be looking at these PLA2, which you can see we get very little sequence coverage, but we get a lot of sequence information from these experiments. So going back to those previous figures, so you can see on the left, we've got the collision-induced unfolding experiment. So here is the drift time on the left and the collision voltage on the drift time on the y-axis and the collision voltage on the x-axis. And if we apply very little collision voltage and still do ECD, we get some fragments that are forming, and you can see that we get fragments that only cover a small portion of the disulfide bond. So we're only really looking at the exterior of the protein. If you increase the unfolding further, now we're starting to get more fragments within the center of the protein, indicating that we are starting to reduce more of those disulfide bonds. And as you go further, you get most of the disulfide bonds being reduced and able to be mapped. So again, we're able to show that we can actually look at all these fragments and hope understand how these disulfide bonds are being formed. We're hoping to take some of this data in the future to then piece it back together to generate 3D structures of these proteins, because these proteins are not well known. For the last little experimental data that I wanted to show you, there is an adage that every Australian animal wants to kill you, even things that are fluffy looking. So what I'm showing you here is a platypus. So platypus is a very interesting Australian animal. It's got a duck bill, it's got flippers, it's a mammal by all accounts. It lays eggs, which is weird. But because Australia provides unique opportunities for these types of species to evolve, you get some very interesting biological things. So platypus, which are native to Australia, have developed spurs and venom glands for defense. So here we can look at the spurs of these platypus. So you can see this is just behind the hind feet here, and there's a spur right there that generates venom. This is only present in the males, and it's really just defense against other males when they're competing for females for mating. And because platypus are mammals, they have a very different mixture of proteins and peptides compared to reptiles. So a lot of venom work previously have been done on reptiles, and also it produces very little venom. And because we're off down in the middle of the ocean, it's not well characterized because there's very few people who are doing these types of experiments. So this work was done in conjunction with the Department of Primary Industries and the University of Sydney. So they've come to us and said, we don't actually know what's in here. So we took the venom, we got some RNA-seq data to generate a protein database, and then did some bottom-up proteomics first. And in the bottom-up proteomics, we got a few different proteins that are present within the platypus. And interestingly, it was this small venom-like beta defensin that the researchers were really interested in, because these are not seen in many other animals, and really it's quite unique to the platypus. So we looked at some top-down experiments to try and map some of that disulfide bonds to try and give some idea of protein structure. So here I'm showing again collision-induced unfolding of this particular protein. So you can see that, well, there's less transitions because there's a lot less disulfide bonds. But if you do the initial experiments, you get a lot of fragments that only really cover one disulfide bond. But if you increase the unfolding, you get access to more of those disulfide bonds, and it allows you to get a lot more information within the center of the protein. And this allows us to get about 38% sequence coverage for this particular protein, as well as really confirm some of these disulfide bonds. And this was the first time, to our knowledge, that this protein was able to be characterized. So I'm just going to finish off a little bit by talking a little bit about the facility. So I work as part of my role at the Australian Proteome Analysis Facility, where I manage the mass spectrometry team. And a lot of the work that we do really is here to help support students and support researchers do these types of experiments, top-down experiments, as well as bottom-up experiments. So I think last time I presented this, there were questions about can we build this into a program that is easily accessible. So I thought I'd just touch a little bit on how we're doing this and how we're trying to make this a little bit more accessible for anyone. So if you've got some sort of interesting experiments, you can probably try and do it yourself as well as build some of the figures that I've shown. So unfortunately, all I can show at the moment is a flowchart because we're only up to here, but I've I've been using MMass a lot for these types of experiments because it's one of the few software packages that really step you through how these types of experiments are done. And it's really been great for teaching students, how to look at their mass spectra data, what it looks like when you deconvolute, how to pull out stuff. However, the previous MMass was written in Python 2 and hasn't been updated for over 10 years. And it's very limited for high charge states because as you try and start to look at things with more than 20 plus charges, it starts to crash. And then if you look at things, you try and use it for native mass spectra, it tends to be quite unable to separate these things out. So we've started by first modernizing MMass to Python 3. So the reason I'm showing all this is because this is how I was able to analyze all our data, and I'm hoping to kind of say, watch this space, we're going to get something out for the community to use hopefully soon. So we've modernized this by updating it to Python 3, and one of the things that has improved is that it's able to now store the data slightly better. So when you do your deconvolutions and things, it actually matches things much better, as well as not crash so much. So it uses a lot more bandwidth. Currently, we're at this stage and it's all command line interface, but we're also building a GUI, so a graphical user interface that should hopefully help everyone be able to use it. And we're also changing the architecture so that it actually sits on the GPU rather than just the CPU, which should allow for faster processing, lower instances of it crashing, and also we want to incorporate the fragment assignment. So currently with MMass, it doesn't quite do that. And we had a tool called Clips MS that I built with Carter and Ben at UCLA that works pretty well. So it should all work natively, and we're hoping that we can just make it into a one-stop shop, as well as put this onto a HTML so it can just be a JavaScript or web-based interface that anyone can get into and use. So the reason for bringing this up was watch this space. I think I've been asked many a times how this data gets generated and looked at, and we're building something that should hopefully help people do that. And lastly, I guess I just got to thank the entire team. So we've got a fairly big team, but within the mass spec team, we've got 5 or 6 people. Ruth, pictured here, is the main person who has done a lot of these experiments, especially for the Venom stuff. And of course, all our funding bodies for funding. And I'm happy to answer any questions.
A fantastic talk, Mohammed. I'm very, fascinated by these very, precious animals and the venom studies. And then certainly, Australia has a lot of unique species in there. So anyway, we'll open up for questions. So Joe already typed his question there, Joe, since you are already, showing your face and just to turn on your mic and speak.
Do you want me to? Okay, I can verbalize it. So,
Yeah.
I just read it. I wasted time. Joking.
okay, this is like group meeting now. Okay,
I'm sorry,
so
everyone.
all right. So, Mohammed, so you showed in your workflow that you had CID following ECD, and I wondered whether the data you showed actually required the CID to fragment the bonds, or was this ECD alone um, sufficient then for a lot of the data?
Yeah, so ECD alone actually doesn't get you any more fragments than what you would think. So if I go back to that just a slide, I guess it doesn't really matter which one I pick. So if, for example, we did this, if you unfold it, you can see that I have the ECD on the whole way, but I have an actual um and some CID. But I think so. There's I guess there's two questions there. So you do not get quite the same amount of fragmentation with just ECD, but if you allow some supplemental energy in CID, it gives you a lot more of this fragment. So I think part of the reason we're doing the CID is while the ECD does help reduce it as well as fragment a little bit, it doesn't necessarily always generate fragments that are detectable. So I think the CID is very light. It's only about 10 volts. So it's only just to knock off any ECD that is, you know, you've got some of the fragmentation but hasn't quite fallen off. So it's just really gentle to try and push the rest of that off, if that makes
But that
sense.
means, yeah, so that means then your internal fragmentites are CZ based, or are they some BY also?
They're mostly BY and we do get some BZs. And yeah, there's not so many CZ internal fragments. They're mostly BY or CY kind of things. So I suspect that the internal fragments are generated due to a secondary fragmentation from the CID that we were including.
Okay, all right. And then my second question, you can read the second question. That's the question I always ask you
Yeah.
is when are we going to see a program that actually outputs the where the disulfide bond locations are
So
derived?
that's part of what we're really trying to chip away at, and I think that's the main thing. So basically, from these experiments, we can really start mapping the change in the reduced disulfide bonds, and by using that, we can then kind of infer where these are. So a lot of these, so actually for a lot of the venom proteins, those disulfide bonds are theoretical as opposed to being
Yeah,
properly characterized.
right.
So a lot of the data that I've shown, which I probably should have mentioned, actually suggests that those disulfide bonds exist. So it's kind of confirmation that you can start doing that, but it's going to take a little bit more work for me to perfectly validate it because, well, I'm a sucker for making sure that everything makes sense first.
Sure. Good, good.
So great question. Thanks, Joe. I want to actually have a follow-up question about Joe's question. So you now do
Yes.
the CID follow ECD. As you know,
Yes.
ECD, particularly as we originally called non-ergodic, and it's a, you have to see the early literatures and also those from Kiki Hawkinson's lab
Yeah.
that ECD generate a lot of fragments, right? So comparison to CID, ECD is non-selective, so you get a lot of fragments. And so that means that each of the fragment, the intensity is going to be low. And so if
Mhm.
you do CID after ECD, I just like this will be even lower when you you get like the low fragment abundance and do the second. So have you thought about switching the order? Do CID first, followed by
Um,
ECD, right? The
yeah,
sequence.
yeah. So I guess I mean, I think if we're really looking at it to maximize how much fragmentation we're getting, that would
Yeah.
make sense. But I think for the purposes of this experiment, what we were really trying to do was to really probe that disulfide bonding and see if we can get from the unfolding, try and
Hmm.
understand some of that disulfide bonding. And I think what's happening with these types of experiments is we're slowly unfolding it, doing the ECD to break accessible disulfide bonds before detecting them. Um, and I guess if I were to do the CID first, then I would fragment it significantly and then do ECD to get me lots of fragmentation. So I would get a lot, I will say, I'll probably get much better fragment spectra because I'll have a lot more intensity. But I think it, I don't know if I can use that method to try and do what we're trying to do in these experiments, which is trying to figure out some of that disulfide bond formation and things like that.
I mean, when you do the ECD of these disulfide, do you see the ECD will be able to cleave the disulfide and get some products? There's a paper from Roman Zubahi when he was postdoc in Pharmacophy's lab. They published a
Yeah.
paper in JACS about the
Yeah.
preferential cleavage on the disulfide bridges. So if that's the
Yeah.
case, that makes sense. You do ECD first, so that can open up the structure by cleavage of
Yeah, and
disulfides.
and, and yeah, that's what we're trying to do. That's what yeah, I guess that would make sense then. So we could I could try it and see what it looks like. But yeah, I think the other reason was because our ECD is currently behind our eye mobility cell, and I was really just trying to use our eye mobility for something fun.
Oh.
Oh, okay. I see Joel unmuted. Joel want to add
Hey,
on?
hey, Mohammed, doesn't this sound
Yes.
like a thesis defense?
Yeah, it does. And it's at 1 a.m., which is great.
Anything else, Joel, you want to say?
No, no, there's some other
Okay.
questions that are on the
Okay.
chat.
Okay. So we have another question from Marika Polak. Is any attempt to do MS3 or MSN of the assigned internal fragment to further validate its internal eye and not a neutral loss side chain loss? Thank you.
Um, so not in this particular project, I didn't, but we have done these types of experiments before on the cyclic because it does give you many, many different places where you can fragment, and we have been able to validate that some of these are actually internal ions. I think, well, I have a paper that's in the archives that I do need to, properly publish, but I think, yeah, we've done these types of experiments and basically, we're pretty confident that they are internal fragments and not neutral losses, sidechain losses, mostly because the math to generate the internal fragments really just match with that. But yeah, for this particular experiment, we didn't actually look at that. So it could be, but um, yeah, I should probably validate that better.
So that's actually is really good question, and that bring up a controversial point for using the internal fragment
Yeah,
and in the complex proteomic samples. I think internal fragment certainly is much more useful when you have a relatively purified sample and no uh, So know that, oh yeah, he has posted another message in there. So,
yeah.
so about the validation, see whether that's really truly the internal fragment or there could be fragment from other possibilities. So I think, especially for the proteomic community, certainly involving these internal ions will bring up a lot more fragments but how
Yeah.
reliable they are. So, can you comment on that?
Yeah, so I think recently, maybe last year, Carol Robinson's group actually suggested that the internal fragments should really only exist if they're anchored by a terminal fragment, which I think is a good idea. Like I think if we're assuming that I guess there's two questions that I might pose back to the community. So if we assume that internal fragments are happening from a secondary fragmentation event, then in theory you should have a terminal fragment that then forms an internal fragment. So you should be able to get, your terminal fragment and then an internal fragment that correlates to an end of the terminal fragment. But if we think that internal fragments just form, then maybe that method wouldn't work, but it would be a way to validate that or to give yourself confidence that those form, because in theory they should be multiple fragmentation events as opposed to single fragmentation events.
Absolutely. And I also posted a paper published by Kiki Hawkinson's group in MCP. From their investigation, they find essentially it's not very easy or very difficult to
No,
observe
it's not.
the
Yeah.
internal eye in ECD. But certainly, the chances will be higher in CID or high energy, dissociation. Likely you just over fragment, and that
Yeah.
cause internal fragment. So definitely still need to be more cautious in there. So
Yes.
I want to read the comment from Marek Polak. And he said, I cannot talk at the moment. I'm at another meeting right now. He's double duty. But I wanted to recommend a paper from Kat Boyker on that he also sent the link. The title is Does Electron Capture Dissociation Cleave Protein Disulfide? Catherine Brooker. No, not Cat Brooker. Catherine Brooker. Yeah. So as I said,
Yeah.
this will be good paper to read. Thanks for the
Yeah.
suggestion. So we now get the question from Paul Dennis. Paul said, "Can you explain what CLIP-MS does and is it available
Hmm.
freely?"
I'm
I
probably
assume
I'm
it's
probably not
available.
the best I'm probably not the best person to comment on it. I think Carter's here to um,
Carter.
listen to me butcher what it is. So
Yeah.
any questions you can definitely ask him later. So Clips MS is a program that I'm not
Here's
going to
Car
bring
here's
Carter into
Yeah.
the spotlight.
Hi, everyone.
Now it's really becoming a group meeting. Carter, here you go. You're on the spot.
Oh no, don't do that to Carter. I can do it.
Now, Carter. So for those of you who don't know Carter, Carter is did his graduate school with Joe, and you wrote, right? You wrote the Clip MS
Right, right.
software and published a paper in JAX, right?
It's in Journal of Proteome Research, published in 2021. And yeah, Clips MS is freely available on GitHub, so feel free to download it. Runs off Python, and you can do the same analysis that Muhammad is doing here.
Yeah, so basically it takes your deconvoluted mass spectra and matches it to terminal and internal fragments. And it's also, I think, on the consortium's website, if I remember correctly, isn't it, Joe?
It should be the paper should be linked there. I don't remember if the program's there.
Yeah. Yeah,
And ho
so
and hopefully we'll see an updated version when Carter goes to University of Oklahoma to start his due faculty
I have
position.
plans. I have plans.
Cool. Congratulations, Carter.
Thank you. Thank you. Excited to start at OU in the fall.
Great. Awesome.
yeah.
All right. So great to know the Clip MS is available. So Carter, for you now is how do you, kind of further develop the Clip MS, like to improve its false discovery or whichever the accuracy for that. Yeah, once you open up to the whole proteome, there's a lot more
Yeah.
as
Right.
possibilities for misassignment and overuse these internal fragment.
Right. Yeah, I definitely plan on, really diving into how these internal fragments form and putting restraints on it so that we get fewer false positives for our analysis. So,
Right.
and of course, developing ways to score these ions is important as well. So I plan on diving into all of that as I become a professor at OU.
Mhm.
And then adding additional capabilities, like if you have multiple protein sequences in a mass spectrum, well, how do you deal with that, right? How do we create better deconvolution algorithms to make sure that all the ions you're assigning are real and are deconvoluted correctly is also important. So there's lots of ways to improve the data analysis to get some accurate assignments.
Excellent. And also, put into these potential just, the algorithms in terms like how possible is these fragment, internal fragment, whether it's, if this is ECD with CID, and also the internal bond preference, cleavage preferences. I think there's quite a lot of fundamental studies on, which amino acid, which bond could be preferably cleaved, and the CID, and, Vicky Wysocki and others have all this information. So, that would be great. Thank you, Carter. And we look forward to your, future independent career work.
Great. Thank you very much.
Yeah,
Sorry for
congratulations.
roping you in, Carter.
It's all right.
Yeah.
You're good.
So if you have not given a proteoform Thursday, maybe you could sign up for the September one.
I think Carter did, didn't you?
Yeah, I think you did.
Yeah,
think you
I
gave
haven't
one.
signed up for anything,
Okay.
but
No, I thought you gave one of these talks before.
no, I haven't given one of these talks yet here at this.
Oh,
Oh,
okay. There you go. There you go,
so
Paul.
yeah, Paul. So see, it's nice, and for everyone in the audience, we would like to invite you to speak to the proteoform Thursday, and if you're interested, just send us your, as I said, contact Paul Dennis. All right. So I think what else we have? Any questions from audience?
Probably should let Mohammed go back to bed.
Oh, okay.
It's go to bed,
Okay.
not go back to
Okay,
bed.
great job,
Oh,
Mohammed.
okay, right.
So another quick thing I was going to suggest, because we did quite a bit of disulfide work before, and I think I find the TSAP is really useful for partial, di reduction. So you could
Okay.
try to open up slowly and you get better fragment. Yeah, we'll let you go back
That's a
to
good idea.
bed. And actually, the last few minutes, if some of you are willing to stay, I'm going to show a few slides from this new women's, top-down women's committee. Certainly, if you guys want to leave, that's fine. But let's thank Muhammad again for his wonderful talk.
Nice job.
Yes.
We should also thank Carter for bailing me out in some of those questions.
Thank you, Joe, for support, and thank Carter for, the stronger support and perspective.
Sure. Now I got to go to
Okay.
school and have a real oral exam. See you later. Bye-bye.
Good to see you,
Yeah,
Joe.
so let me see what I could share this one. If anyone's still there, I'm just going to quickly introduce. Some of you already see that in the social media, we now have this new committee in the Women in Top-Down Proteomics Committee. So chaired by Professor Si Wu in University of Alabama and also Professor Taro Pakala from Adelaide University, also from Australia. And I serve as a liaison from the board to this committee. Let's see. Oops, got a lot. Yeah, so we have a representative from industry, from postdoc, and from national labs, Carolyn and Huiling from China, and Carolyn Liu from Florida University, and Corinne from University of Oxford, and Elizabeth from Merck, and Kelly from Alabama. So I think they quickly put together a really beautiful plan and to show that this Wisconsin Women's Committee is affiliated with CDTP, but a dedicated foster network, mentorship, professional development, and scientific collaboration among women scientists and top-down proteomics. But just a disclaimer, you can also join you're not a woman, but this is really open up to women and the supporters. The mission is to create an inclusive and supportive environment that connect researchers from academia, industry, government, and around the world while advancing scientific collaboration, visibility, and opportunity. Just quickly to show that, yeah, there's already a lot of pillars. The mentorship of professional development is the key, and this responsibility, they move up very quickly. And we already have these first meetings, and they have well, the membership. So it's free. So I'm just trying to call for everyone's attention. So many of you already joined. If you haven't, please join us. And this will be a really good supporting group to support women in top-down proteomics. Yeah, so they already have proposed activities. So pay attention to the announcement in social media. All right. So here is please, join this top-down women's community and join, connect, and network. And this allows you to connect with top-down proteomics researchers and join the discussion and stay updated. All right. If you have any questions about this new women's committee, please let us know. Okay, without further questions and comments, I think we will just conclude this Proteoform Thursday. Thanks again, Mohammed, for his exciting work. We look forward to hear more on this disulfide study.